Chapter 12, Section 3, RNA and Protein Synthesis. The double helix structure explains how DNA can be replicated or copied, but it does not explain how the genes work. As you will see, genes are coded DNA instructions that control the production of proteins within a cell. The first step in decoding these genetic messages is to copy part of a nucleotide sequence from DNA into RNA, or ribonucleic acid, that is. These RNA molecules then carry out the process of making proteins. The structure of RNA. RNA, like DNA, consists of a long chain of nucleotides. As you may recall, each nucleotide is made up of a 5-carbon sugar, a phosphate group, and a nitrogenous base. There are three main differences between RNA and DNA. The sugar in RNA is ribose instead of deoxyribose. RNA is generally single-stranded, and RNA contains uracil in place of thymine. You can think of an RNA molecule as a disposable copy of a segment of DNA. In many cases, an RNA molecule is a working copy of a single gene. The ability to copy a single DNA sequence into RNA makes it possible for a single gene to produce hundreds or even thousands of RNA molecules. Types of RNA. RNA molecules have many functions, but in the majority of cells, most RNA molecules are involved in just one job: protein synthesis. The assembly of amino acids into proteins is controlled by RNA. There are three main types of RNA. Messenger RNA, here, ribosomal RNA, the stuff that makes up the ribosome, and transfer RNA. The structures of these molecules are shown in figure 12-2. Here is the transfer RNA, guys. It transfers amino acids, to the ribosome. Most genes contain instructions for assembling amino acids into proteins. The RNA molecules that carry copies of these instructions are known as messenger or mRNA because they serve as messengers from DNA to the rest of the cell. Proteins are assembled on ribosomes. Ribosomes are made of several dozen proteins as well as a form of RNA known as ribosomal RNA. During the construction of protein, a third type of RNA molecule transfers each amino acid to the ribosome as it is specified by coded messages in the mRNA. These RNA molecules are known as transfer RNA. Transcription. RNA molecules are produced by copying part of a nucleotide sequence of DNA into a complementary sequence in RNA, a process called transcription. Transcription requires an enzyme known as RNA polymerase that is similar to DNA polymerase. During transcription, RNA polymerase binds to DNA and separates the DNA strands. RNA polymerase then uses one strand of DNA as a template from which nucleotides are assembled into a strand of RNA. A process of transcription is shown in figure 12-14. How does RNA polymerase know where to start and stop making an RNA copy of DNA? The answer to this question begins with the observation that RNA polymerase does not bind to DNA just anywhere. The enzyme will bind only to regions of DNA known as promoters, which has specific base sequences. In effect, promoters are signals in DNA that indicate to the enzyme where to bind to make RNA. Similar signals in DNA cause transcription to stop when the new RNA molecule is completed. So here, guys, we have a picture of the nucleus, and inside the nucleus, as we talked about, you have DNA. You have the enzyme RNA polymerase that binds, that means just connects to the DNA, to one side of it, and then it starts copying that segment, that it's going through. And remember, this RNA, this messenger RNA here, Okay, it's basically a copy of the other side of the DNA molecule, just like we talked about in class. Okay, so make sure that that is clear and that transcription, okay, is very, 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 very clear. If you have any more questions about this, make sure that you ask during class. Okay, remember that uracil is not found in DNA, so the base pairing rules are a little different. Okay, so when it sees a adenine on the DNA strand template, it'll add a uracil, okay, when it's synthesizing that mRNA molecule. All right, hope transcription is a little clearer today.